Good afternoon and welcome everyone to our GCSE Science Revision Session 2 today. Great to see so many of you coming along and joining again from last time as well. So just an overview of what we're going to be looking at today. We'll be looking at practical skills, graph skills and analysing data for paper 2. Today I'm joined by Neelam, Lakshmi, Jess and I'm Claire. We're 4 of the science teachers here at Academy21 and we're going to be going through some of the top tips from us and the other examiners in our department as well to help you do your absolute best in the upcoming paper 2 exams for science. So first, a few housekeeping things. Can you double check you can hear us? I really hope you can hear me, and please thank you for filling in the polls there. You'll see a little poll just down to the side of me. Let me know if you can hear us, and if you don't hear me right now, I hope you're pushing no and you can tell us in the Q&A and we'll try and help you out with that. As well as that, if it's helpful to you, we do have closed captions available, just kind of up there above Jess and Lakshmi, there's a little CC button. And if you push that, you should be able to turn those on and follow through if that helps you as well. We're also going to have quite a few polls today. We've got the one just now and we'll have some other ones to get you thinking about the examples that we're looking at as we go through today's session. And we have some files next to me here that you can download. There is our revision planner that some of you may have from last time, and you can use the paper 2 sections of that to see the different topics and information that would be in there. We have also got the file from today's revision session. That's file number 3 there. And if you're here today as well and you want your attendance certificate, please do remember to download that before you leave at the end. Throughout the whole session, we'll have different science teachers leading different sections of this, and the rest of us will not have our cameras on, but we'll be in the background answering questions in the Q&A. So please do ask any questions as you go along to do with your paper 2 science exams or anything that we're going over today. So we are going to start off by looking at practical skills, and for the practical skills today, I'm going to hand over to Jess. So thank you very much, Jess.
Great. Okay, so we'll have a look at some of the practical skills then. So on the combined science course, there are 21 different required practical activities. So you'll be asked questions on some of them and about their methods, but you might also be asked about ones that you've not seen before. But with those ones, you don't need to panic. You just need to apply these same practical skills to those different practicals and those questions as well. So we'll have a look through some of the skills then that we use for these practicals. So starting off with a hypothesis. So a hypothesis is a statement that can be used to test, to confirm whether or not a variable has an effect on another variable. So basically a hypothesis is a statement which can tell us what relationship there is between different variables. A prediction is what you think might happen. So, for example, if I was to say, I think that if I double the weight on the spring, the extension will also double. That would be a prediction. The next thing then is to actually do the experiment. So whatever experiment it is, you need to go ahead and do it. And this experiment, remember, is to test your prediction that you made beforehand and also collect some results. Once you've collected those results and you've done your experiment, you can then go through the stages of analysis. So with analysis, it's just checking that those results are valid. Can they be compared? Have you repeated them? Maybe calculated an average just through your results there. Okay, and then the final step we have here is the conclusion. So from your analysis, you can have a look at the results and see whether they agree with your prediction and your hypothesis. And they can show us as one variable increases, what happens to the other variable. Does it also increase or does it decrease or something else? Okay, so that's sort of the main kind of skills associated with practicals. Speaking of variables is one of the most common questions they can ask you about practicals. So in science, there's 3 different variables. So if you have any ideas for what any of those variables are, feel free to let me know in the Q&A, and then we'll go through those and their definitions and identifying them in just a moment. Yeah, great. Thanks for popping some in the Q&A then. Brilliant. Okay, so those three different variables then, the first one is the independent variable. Okay, so in the poll, feel free to let me know which of those definitions do you think best matches the independent variable. So feel free to let me know in the poll. You can guess if you're not sure, or in the chat as well if you'd like to. Yeah, brilliant. Well done. Yeah, great. Okay. So the independent variable then is the variable that you are choosing to change. So it's the one that you are changing, or the one that you want to see what effect that variable will have. That is the independent variable. The next one then is the dependent variable. So again, feel free to let me know in the poll or in the chat, what do we mean by a dependent variable? Yeah, brilliant. Well done if you voted. Yeah, lovely. Well done. Great. Okay, so the dependent variable is the variable that you measure. So the one that you would write down as your results. So for example, if I wanted to investigate what type of shoes or how types of shoes might affect my running speed, the dependent variable would be my running speed. So the actual results from my experiment. Finally, the last one then is the control variable. So again, feel free to let me know in the poll, what is a control variable? Yeah, excellent. Well done in the poll. Brilliant. Okay, so a control variable then are, these are things that you keep constant or keep the same. And the reason we do this is to keep our results valid and mean that it's fair to compare them. Okay, so for example, going back to my experiment again with the types of shoes, if I was going to run in different types of shoes, control variables might be things like the weather, maybe the type of breakfast I've had, even the runner who's actually running in the different types of shoes. So things that we need to keep the same in order to have a valid comparison. Okay, so I'll just pop those all up on screen for us. We can see the definitions next to each of those variables. If you really struggle to remember the difference between the dependent variable and the independent variable, you can use this down here at the bottom, ChinMed. So hopefully you can see that down there at the bottom. And what this means is, well, chin is for change and independent. Okay, so the CH is for change, the start of change. IN is for independent. And then med, the ME is for measure, and the D is for dependent. So if you get kind of confused or mixed up between the independent and the dependent variables, you can just use ChinMed, and that can maybe help you remember the difference between them. Okay, so what we'll have a go at next then is we'll have a look at an example of an experiment and see if we can identify the different variables in this experiment. So this experiment then is to investigate the effect of light intensity on the rate of photosynthesis using an aquatic organism such as pondweed. So take a moment just to think about it and feel free to let me know either in the chat or in the polls there. What is the independent variable, so the one we're choosing to change, and what is the dependent variable, so the thing that we are measuring? So feel free to have a think about it. Let me know if you have any ideas, and then we'll go through it in just a moment. Yeah, brilliant. Well done if you've popped some ideas in. Yeah, lovely. Well done. Great. Okay, so the independent variable then, so the thing that we are changing or choosing to change, this would be the light intensity. Okay, sometimes it helps as well, especially when they're written like this, the independent variable is usually what follows effect of, as that's the one we want to see what effect it has. Okay, so the dependent variable then, remember this is the one we're measuring our results for the experiment. This is going to be the rate of photosynthesis. So this is what we'd actually write down as our results. This is what we're measuring. That would be our dependent variable. Okay. Next thing then we have then is to have a think about some control variables. So if we're looking at the rate of photosynthesis, what might be some things we want to keep the same? So some control variables. So feel free to have a think about it. If you have any ideas, let me know in the chat as well. And then we'll go through some of them in just a moment. So any control variables, things we need to keep the same. If you're struggling as well, feel free to use the diagram to help you. That might give you a bit of a reminder. Yeah, lovely, well done. Great, okay, so some things that we might want to keep the same in order to keep our results valid could be things like the amount of water, the temperature, and the carbon dioxide levels as well, okay? So control variables, you keep them the same to keep your results valid and allow you to compare them as well. Okay, so the last sort of thing then that we'll look at in terms of practical skills is the equipment. So with the equipment, you need to be able to identify it and also to be able to draw it as well as part of a equipment setup. People often panic when they are told to draw these pieces of equipment. It doesn't need to be anything fancy. It doesn't need to be like 3D or shaded in, anything like that. It just needs to be the outline. And if you're drawing them as well, you should label them as well. So even if it doesn't look perfectly like it, you should roughly be able to tell what it is. Okay, so with these pieces of equipment then, I'll just pop up what they actually are. So this first one we have here then is a Bunsen burner. Again, this one looks a little bit complicated in terms of how it's drawn. It doesn't need to be especially detailed, but as long as it's labeled and it looks roughly like it, then it should be fine. A beaker is this one here, measuring cylinder, and then a test tube. Underneath that, there is a funnel, a clamp stand, and this one here, people often forget, is the conical flask. So a conical flask has like a narrow neck and then it expands out at the bottom there, okay? So as long as you label them and it looks sort of roughly like it, then you'll be absolutely fine with those, okay? So that brings me to the end of the practical skills then. So I'll pass on to Neelam for the graph skills.
Hello everyone. So now that we have taken a look at some of the practical skills with gestures there, we're going to be looking at how, we're going to try to consider how we may best present those results that we might have collected, the data we might have collected in the best way possible using some graph work, okay? Very, very good skills to have under belt if you're going into an exam situation, because it's worth easy marks on an exam and very, very easy to get those marks. So let's have a look at some of those now. Okay, so we are going to be taking a look at one or two examples first of all. The first example is this one here, using the practical that Jess was talking about a couple of minutes ago, where we're looking at the rate of photosynthesis and how it is affected by the intensity of light. So this is a typical set of data that you might have collected from a practical such as that. You'll probably be given this data on an exam question, and you might be asked to plot that data onto a graph. Now, first thing we've got to consider is that what kind of graph are we going to draw, okay? So because this data is continuous data, the first thing we must remember is continuous data is always a line graph. So we're going to be plotting points, not bars on this graph. And we are going to use small crosses in order to plot the data onto this graph. Remembering the equipment that you need to take into an exam in order to plot graphs, make sure you have got a sharp pencil with you and a spare. Make sure you've got an eraser with you also, and also a ruler just in case you have to draw a straight line as well. Okay, so going to have a look at how that might be plotted now, and then look at any correlation between the distance of the lamp and the number of bubbles of oxygen gas that would be collected in this case. So going to plot some data on now. Here's one I prepared earlier. So here we go. So plotting some data on. So using crosses rather than dots is the best way to plot our points, because if you use crosses, you're more likely to get a more accurate pinpointing it exactly where it's got to go, and it is better than putting a dot, which might look too small, and also sometimes it could be too big if you have not got a sharp pencil. So plotting on the rest of those points using small crosses, and clearly we can see that there is a correlation in the data, okay? So now we're going to consider what the correlation is. Of course, the next thing here would be to draw a line of best fit, but we'll take a look at lines of best fit in a couple of minutes, okay? But just looking at this data now, is there a correlation between the data? Okay, so we can clearly see that there is a correlation here between the data that we can see, okay? So here the data is showing us that there is a negative correlation in the distance. You know, we're saying that as the distance increases, the number of bubbles decreases, okay? It's not a linear correlation because you should be able to see right towards the end of that graph, it's curving off slightly, okay? So it's not linear, but it is a negative correlation. So saying that as the distance from the lamp increases, the number of bubbles produced is decreasing is quite important there, okay? So you can see a correlation straight away. Now, another example of a graph. This one is a physics example this time, okay? So this is the Hooke's law example. So a student investigated how adding masses to a spring affects its extension, and they put their results into a table. So the table here is showing force in newtons and then the extension of the spring in centimeters, okay? So there is a set of axes there ready drawn for us, but we might have to consider what is going to go on which axis. Now, the rule generally is that the independent variable always goes on the x-axis, and the dependent variable, that's the one that we are measuring, always goes on to the y-axis. Okay, so here in this case then, could you please let me know which axis should be the x-axis in the poll, please? You'd let me know which one is going to be the x-axis and which one's going to be the y-axis. If you could let me know in the poll, please, which one is going to be which in this case. What's going to go onto the x and what's going to go onto the y. Very good, well done. Good, well done. So the variable which is going to go into the x-axis is the one which is on the left-hand side of the table. The force, which is our independent variable, is going to go into the x-axis, and the y-axis is going to be the extension of the spring. Very good, well done. Well done. Okay, so when you presented the data, you may then be asked on an exam to describe the relationship. You might be asked one or two different types of question here. One could be describe the relationship between the mass added and the extension of the spring, or you could be asked describe the effect of adding the mass on the extension of the spring. Really very, very similar questions, but all you've basically got to do is you've got to say what you think is the relationship between the two of those things, okay? So looking at that, we could say that as the force increases, force in newtons increases, so the extension of the spring also increases, okay? So simply here, what we're saying is that we are just basically saying how this increasing affects this, okay? So it's how the force increasing is affecting the extension. So you don't have to make up words here, just look at the axes that you've got and use that to help you to build your sentence. So you can say, as the force in newtons increases, so the extension in centimeters of the spring also increases. Now we come to the important part, which is going to give you an extra mark, okay? You should be able to see that after up to 10 newtons, we've got a nice straight line, okay? After 10 newtons, is starting to level off. Now, if you can actually mention that on your answer there, you are going to gain an extra mark for saying that, okay? So use the graph and actually mention where the change is taking place. It's quite important that you do that to pick up an extra bonus mark there, okay? Now, as I said earlier, we're going to be looking at how we can draw lines of best fit as well. Now, very, very important that we remember, there is a misconception that lines can only ever be straight, okay? Lines can be straight lines and lines can also be curved lines. So make sure that when you're drawing your line of best fit, you look at the data that you've got in front of you, the points that you've got in front of you, and look at them and decide, does it look like generally a straight line or does it look like a curved line? If it looks straight, then it probably is a straight line. So make sure that you're using a ruler in order to draw it. There may be anomalies. There may be lines, points which are not fitting on the line. That's absolutely fine. You draw a line generally in the direction that the points are going, okay? So in this case, the first one here is a straight line, okay? So you would use a ruler in order to draw that line. Yes, you have got an outlier just here, an anomaly just here. And all of the points do not have to sit on the line. This is not a dot-to-dot drawing. This is a line of best fit. In other words, it's best fitting through the points. So you draw a line through the points. With some points may sit on top, above the line. Some may be slightly below the line, okay? And the second graph, you can see very clearly, is a curved line there. Now, there are many people I've seen when I've marked papers before, when they're drawing their curved lines, they still draw it with a ruler, okay? And they're drawing a dot to dot. Your curved line should be a smooth curve, okay? A smooth curve. So it's like you have to draw it freehand, have to draw it with a sharp pencil, and it should be a nice smooth curve. Okay, I often find that drawing a curve like this one here is best to turn your paper actually upside down, because drawing a curve like that is much easier than drawing a curve like that. So just think about the way to draw it and use a fine pencil in order to do that, okay? So you may have, as I said, straight line graph, or you may have a curved curved line instead. What we have to do then often is we have to use the line that we've got, or the results that we've got from the graphs, in order to draw conclusions, okay? So this is the point where you've got to draw a conclusion by looking at the line graph, or the line that you've got in front of you. So you've got to look at that and decide, what is it telling me about the practical that we were going to do? Now this is going back to our biology practical again. So the distance from the light source and the number of bubbles produced on the y-axis. So again, remember what I said earlier, when you're drawing a conclusion, we say as the distance from the light source, then you can say what happens to the number of bubbles produced, okay? So this is an example. As the distance from the light source in centimeters increases, the number of bubbles produced decreases, okay? So that's showing us that we've drawn a conclusion by looking at that. Now looking at this particular next graph, and be ready because I'm going to be asking you to give me an answer for yourself now, okay? So here we have got a current and voltage graph. So this is now an Ohm's law graph from physics. So you've got voltage and you have got current. Could you try to write your answer in here? Can you write your conclusion for this, please? See what you can come up with. See what you can come up with. Can you write a conclusion for this one, please? Very good, well done. I've got a couple of answers coming in here, fantastic. Okay, so remember we're using here, as we say, as this one increases, this is what happens to this one. So always start with the x-axis as, very good, well done. So as the voltage increases, the current also increases, well done. Okay, folks, so now we're going to have a look at a slightly different type of graph, okay, very similar to what we've already done, but it might look quite scary in an exam situation to us, okay? So this type of graph here, what you've got is you've got your time given to you, okay? This is from a rates graph from a chemistry required practical now. So you've got the time in seconds and you've got the volume of gas produced for using 2 molar hydrochloric acid, okay? And then 1 molar hydrochloric acid also, okay? So you the volume of gas produced for both of those in centimeters cubed. You've got both sets of data given to you, so you are expected now to draw 2 sets of data onto the same graph, okay? So, and then look at the results and look at the graph and then come up with your conclusion. So here it's really important that you don't get yourself confused, okay? And make sure that you're remembering that you have got a set of axes to draw on here. So you've also, as well as putting on your numbers, you've also got to put the units in place, okay? The time is in seconds and the volume is in centimeters cubed to make sure those units go on to your axes as well. So let's have a look at the graph which has been drawn, okay? My suggestion to you here, my advice to you here would always be, don't go and draw both at the same time. Draw one set of results first, draw your line of best fit through those results and label your line. Then do your second set of points, your second line of best fit, and then go on and label that line. So you can see clearly the two different ones. I've seen often, sometimes people have drawn all their points and then they can't see which set of points correlates to which line, okay? So it's worth usually around 4 marks. So it's really important that you get to do this, okay? Because it is really, really big marks for you, okay? And I think graphs are always a gift in an exam paper because this is the time where you can use your skills that you haven't had to memorize, okay? So it's well worth taking your time. Remember, sharp pencil as well. Right, here is the graph drawn, okay? So you've got, as I said, 2 sets of results. So you've got the first one for your 1 molar hydrochloric acid, okay? And you've got your second, and remember, you've drawn it, you've drawn the points, you've drawn your line of best fit as well, and then you have labelled it as well. Then you go on to do your second set, you've drawn it, put your points on, drawn a line of best fit, okay, and you've labelled it. Now you can clearly see both of those values, or both those lines, one for the 1 molar and one for the second. Now you can look at that and notice here that the labels are on: time in seconds and volume of gas. It's not actually been missed off that axis there, but volume of gas centimeters cubed, okay? And you put your numbers on very, very clearly there as well. Right, so now that you can look at this graph, you can see, you can answer the question, is there is the rate of reaction faster for the 2 molar acid than it is for the 1 molar acid? Can you give me an answer just in the Q&A, folks? Do you think that's a yes or do you think that's a no? Is it faster for the 2 molar than it is for the 1 molar? What do you think? In the Q&A. In the Q&A. Is it faster for the 2 molar? This one's the 2 molar, the green line. Or is it faster for the 1 molar? Well done. Yes, it is faster. It's faster for the 2 molar acid. How? How do we know? Okay, we know because the line of the green one, the green line, the 2 molar line here, okay, is much steeper at the beginning. That's the initial rate of reaction. Much, much steeper at the very beginning of the reaction. That means steeper. Another word for that is the gradient is steeper. Okay, that means that there's much more gas released every second for the 2 molar hydrochloric acid, which means it's a faster reaction. Okay, and notice that one is much, much steeper, faster rate of reaction. That one's not as steep, the gradient is not as big, so that means it's a slower rate of reaction. What you should notice though is both of them are finishing around the same height. Okay, that is because it's the same total volume of gas is the same for both of them, okay? The same amount of gas is released for both of them, same volume of gas is released for both of them, but one is just released much slower than the other one, okay? And why is the same amount of gas, same volume of gas produced? It's because you are using the same volume of the hydrochloric acid to begin with. That would be one of our control variables. You would use the same volume of hydrochloric acid at the beginning for both of them, and the only thing you would change here is our independent variable, which is the concentration of the hydrochloric acid. Okay, right, so there we have looked at today how we can use graph skills to present our data so that we can more clearly see a correlation in the results. Okay, now we're going to be moving on and Claire is going to be talking you through how we can analyse the results.
Thank you, Neelam. That was really helpful, and it is really important, and we're going to touch on it and link it a little bit to data analysis here using our graphs. And it is such a great way to be able to pick up marks because it doesn't always involve, like Neelam said, learning and memorizing everything. So now we're going to look about how to use data that we're given and to analyze that data, which can be some really solid marks that you're going to get in many of your paper twos. But first of all, a little bit of vocabulary to start this section tonight, okay? So there are 3 types of averages that you'll have learned about, both in maths and also with us in science. There's the mean, the median, and the mode. And a lot of the time, most of the time in science, the mean is the one that we use when we've got a set of data and we take an average by adding all the values together and then dividing by how many values there were in total, giving us what we sometimes just refer to as an average, but that's actually the mean. It's a type of average called the mean. The other one that commonly comes up in science papers is working out the median. And the median is the type of average that you get by writing out all the values in order and then just choosing the value that comes in the middle. And we're going to see an example of that one in a moment as well. The least common one that doesn't come up very frequently in science, but you will have seen in maths as well, and in some science examples throughout your learning, is the mode. And the mode is just by finding the value that occurs most frequently, so the most common value as well. Now, the other 3 words that are in this table, valid, repeatable, and reproducible, are often talked about together, but they have quite clear and distinct definitions. So a valid experiment, sometimes referred to as a fair test, is an experiment that's designed in a way that controls all the variables that should be controlled. Okay, so it makes it fair. And we very carefully measure all the variables that need to be measured. And we also change very clearly the one that we're changing. This is linking back to your work on variables earlier on in tonight's session. So with an experiment that's valid is a fair test. It's valid. It can answer a proper question in science. If you wanted to know what is the effect of increasing concentration on rate of reaction, then you would design an experiment that allowed you to answer that question with all the variables fixed. We then have repeatable. So an experiment that's repeatable is one that we could do. We do it once, then we do it again, and we would be able to get the same results if we do it in the same way again. Okay, so that's repeatable. You can repeat it. It does make sense. But the other word is a similar word, reproducible. That means that effectively someone else can repeat it. So you could write your instructions for this particular experiment and somebody else could do it following those and get the same or very similar results. Or you could do the same experiment to answer the same question, maybe using a slightly different method or different equipment, or even sometimes different reactants, but it would still answer the same question. That's something that's reproducible as well. So that was our language that we use around these. But something for you to do. I'm going to move to a poll and get a bit of thinking for you just now. And you absolutely can use a calculator if you want, always in science, okay? Though you might not need to for this. So we're going to look at some data that's taken from one of the required practicals in science. And this is the one that's ecological sampling. So it's when you take a square called a quadrat and you throw it out in a field somewhere, usually. And the examples that we've learned about at Academy21, we've looked at counting daisies or dandelions in a big wide field. You throw out the square, it drops down in a random position that you've thrown it out quite often. So it's called the quadrat, 4 sides. So they've placed the quadrat 10 different times in different areas on the school field. It says that the results are shown below. And you can see in this table, the top line in the table is the quadrat number. So like sample 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. They then looked at how many dandelions in each of the samples were in the shade and put them in this line, and how many dandelions in each of the samples were in the sun. So my first question for you, and it's, I see some are already having a go, but it's what is the mean number? So thinking, how do we calculate mean? The mean number of dandelions in the sun. Okay, so that's this bottom line here. Can you give me a numerical value just by typing it into the poll over there for the number of dandelions in the sun? There's a very good chance that you are going to be asked in one of your exams to calculate a mean, and it's just like this that you're going to do it. Calculate a mean, yes, absolutely. So yes. And if you're not quite sure, or you're thinking, oh, that's a lot of numbers to add in my head at this point in the day, you can use a calculator, or just tell me in the little poll box there, you can type in what you would do. What would you do? How would you work that out? Good, good. Okay, so I'm going to go through my working for this, but you can still be putting your numbers in as you go as well. Yeah, good answers coming through. So I've done the shade one first, and I'm just going to run you through that, and then I'll give you a little bit more time to have a go at the sun one. So if I was doing the mean for the dandelions in the shade, I'd go back to my method that I have, which is adding together all the numbers in that row. So all these numbers here, I'm adding them all together to get a total value. And that total value is 35. I'm then going to divide that number by how many entries there were. And in this table, there was 1 to 10. So I divide it by 10, and the answer is 3.5 for dandelions in the shade. I asked you about dandelions in the sun, and we've had some good answers coming through. Well done there. And you can see, actually, everyone that answered was correct. So very, very well done on that. Fantastic. So we're adding those together, dividing by 10. Okay. I would recommend, and you might think, oh, I don't need to do that, but please do write your working for this. Even though it might be a relatively straightforward sum that you're doing, it's really easy under exam pressure to mistype a number or to just copy down something wrong. But if you've written out this working, you might get some marks in this. So please do make sure you're always writing out your working, dividing by what you've got, and you'll get your final answer. So that was a mean. Let's have a look onto having a think about a median. And I'm just going to do this one for you and show you the method that we would use. Using the same data, okay, so this is the exact same data from the top. But we said that when we want to calculate a median, we need to write all the values in order. Okay, so in ascending order from the smallest to the biggest is what we have here. So this line here from the table at the top, dandelions in shade, has been written out again, showing us the smallest number all the way up to the biggest number. And notice where there's a repeat number, we write it as many times as it appears in the top column. We then have done the same here for dandelions in the sun. So we are then looking for the middle value. Now, sometimes when you do a median value, there's an odd number, like say there was 9 results, then it's really easy to pick the middle one because there's 4 numbers and then the middle number, then another 4 numbers. In this case, your midpoint is between 2 numbers. So that's when we have to use the second step that I've got in brackets here. If there's 2 numbers, you take the mean between the 2 numbers. So in the case of dandelions in the shade, there's just 4 because it's 4 and 4 are either side of the midpoint. But for dandelions in the sun, we have 5 and 6. So the midpoint, the median value for dandelions in the sun there is going to be 5.5. Okay, well done on having a go at that one with the poll, everyone. It's nice to see you all joining in today. So I do have a bit more vocabulary for you, but it's really good to get your mind really clear on what these mean going into the next set of exams. So when we're analysing data, we're thinking about various aspects of it. And first of all, we think about, is it accurate? So if data is accurate, it means it's very close to the true accepted value, to the value we would expect. So let's say we were measuring the boiling point of water. We know that the real value should be 100 degrees Celsius. And if we do our experiment really accurately, we would measure it at 100 degrees Celsius. But if it was less accurate, it might measure it a little bit above or below. So it's how close it is to what the actual answer should be. So that's accurate. However, we've also got the word reliable. So if we are able to repeat a scientific investigation and reproduce it and get the same data, we're trying to make it more reliable. So doing more repeats makes it more reliable. This is a really popular thing to come up if you're asked to suggest how you could improve an experiment. The top answer is often just do it again or do it more times so that we can take an average. It's a really good answer to put. That's going to make your experiment more reliable because you're not just relying on one set of data that could have had a random error in there as well. We also have these 2 words, similar but not quite the same, that we mentioned earlier. Repeatability, remember that's when the same person or team can repeat something under the same conditions. And we have reproducibility, where it's possible to transfer that to slightly different versions of the experiment or for other people or teams to do as well. And I really like that, the little kind of target diagram visualization there to give you a kind of visual feel for the difference between these words. And I'll remind you again at the end, but when we have visuals like this, you can download the slides from today's session over at the side. It's number 3, just over to that side of me, if you want to take a copy of any of these pictures as well. So we're going to go on to a few bits with some more polls in here with mine, because we're going to think about how we can do each of the things that we have talked through today. So I'm going to start off by going to this one. Yes. How can we make an experiment more reliable? Can you tell me, have you learned that this afternoon in the session? How could we make an experiment more reliable? I'll give you a moment to think on your answers to that. And remember, while I'm asking these, if you have any other questions, the team are still there in the Q&A. I see them answering your questions, but if anyone else has something they're wanting to answer, do start popping them in there as well. So making it reliable. Yeah. Good thoughts there. Okay, I'm going to move us on. Absolutely. Repeats. You say that, you've got your mark. Do it again. Do it several more times. Which of these is the correct definition of repeatable? This is an easier poll. You can just click a button. So which is the correct definition? There's only 2 choices. Repeatable. Click the one you think it is. Brilliant. Well done. Oh, 100% of you are correct so far, so don't change your answer. If you haven't had a go, have a click. Very well done. I'll just show you the results there. You're absolutely right. The same person repeating and obtaining the same results under identical conditions, which means if I go over to our last definition, let's see if you can get that one as well. And we'll be 100% twice. Can we say what it's going to be for reproducible? And this will help you remember the difference between both of them. Absolutely. Yes, well done. We got that one. Brilliant. Okay, and again, we had 100%. Absolutely fantastic. That's the one where it can be done again a slightly different way or by different people, and it'll still be reproducible. One final slide I want to show you, and then we'll just go for any final questions. This links back to the stuff that Neelam showed you about rates, but I always like to mention, when you get a graph with a rate curve on it, there's 2 important things on that curve, not just one. The first important thing is how steep it is. And I'll ask you this question and just pop in the Q&A. Where is it steepest? Anyone tell me, where is it steepest? Not where my pink arrow is pointing. Where's the absolute steepest bit? If that was a hill that you were climbing, what's the worst part? Yes, it is. It's right down there at the very start. All rate curves are steepest at the beginning and then level off, and that is because that's when there's a maximum concentration of reactants. As it levels off, that means it's slowing down. When it gets completely flat, that's where it stopped. And that's the other interesting part on this graph, because where it levels off, that tells us something too. That tells us how much of a certain reactant there was, the limiting reactant. So that's important too. And the final thing I'm going to ask you before we just go to final questions, if I asked you, this is an experiment where we've made some gas, okay? If we heat this experiment up, we heat it up to a hotter temperature, what would the curve look like compared to that one? So we're going to take the same reactants, the exact same amount, same volume, same concentration, but we're going to heat them up to a hotter temperature. What would be different about that graph? And if you got the question, sketch on another line to show it, what would you draw? You can just tell me in the Q&A. Yes, good thoughts there. Somebody got it. What I'm getting at there is if it is hotter, that's a factor that affects the rate and it's going to make it faster. It will go up steeper. Okay, so we have this steeper curve, but I said all the other things are the same amount of substances. So the final amount of products produced is going to be the same just at the end there in this part here. Okay, thank you for listening to that final section. I think the others are going to just pop back, and if you've got any questions for any of us, you can pop them into the chat as well, the Q&A chat, sorry. These are just some of our top tips based on what we said today. Remembering the chin mid that one of us introduced earlier, just saying a change is the independent variable, that's the one we change, and measure is the dependent variable, the one that we take results for. Remember that your x-axis is always the independent variable that you have been controlling in that experiment, and y is the one you're measuring, the dependent variable. Always show you're working for everything, including averages, and basic diagrams like you saw with Jess in the first part we're not artists in science. We just need to do a clear two-dimensional representation when we're drawing something. And please do link your variables to the direction of change. So if you say, as the temperature increases, the rate of reaction increases, that's showing the direction of change as well. So thank you very much to everyone for coming along today. We want to all wish you the best of luck for your paper 2s, which are coming up in the next couple of weeks for biology, chemistry and physics. We're still going to be here for just answering your questions in the chat as well. But it was lovely to see everyone this afternoon. So thank you for coming along. And do remember to download your attendance certificate before you go, as well as the other useful resources that we've popped in that box for you too. Thanks very much for coming along, everyone. Bye-bye.